All right, so we're picking up where video 1 on cellular respiration left off. So we had talked about the 3 steps. So we have glycolysis being step number 1, the Krebs cycle, and electron transport chain being the 3rd step in aerobic cellular respiration, so oxygen requiring cellular respiration. So the Krebs cycle, we talked about in glycolysis, glucose is going to break apart from a 6-carbon chain into smaller 3-carbon chains called pyruvates. Those travel into your mitochondria where they're broken down. So we lose another carbon and we're going to turn it into citric acid, and then it's going to enter the Krebs cycle. Now, once it's in the Krebs cycle, those carbon chains are going to continue to be broken apart. So again, the big idea here, bonds are stored energy. So as you break bonds, you release energy. And so as those carbon bonds are broken apart, the cell is going to use that energy that's released to make ATP. It's also going to store some of that energy in high energy hydrogen bonds on your NADH and FADH molecules. And these NADH and FADH molecules are going to travel to the electron transport chain where they'll be used later. Now you'll notice that we've been breaking apart a lot of carbon. So we started with 6 carbons, and now once we've gotten through the Krebs cycle, we've broken all of our carbon chains down. Now, all of those carbons are going to come across as waste. So as we break apart glucose, this is where that CO2 comes from, so carbon dioxide. So remember, you're going to take in oxygen as a part of doing cellular respiration. So as you break off carbons, you're going to stick it on that oxygen and make carbon dioxide, which is going to be your waste. So that's where those carbons come from. Now, don't panic. Here's a detailed look at the Krebs cycle, but I just want to give you guys the big idea here. So here's acetyl-CoA, which is also citric acid, so right there. And that's going to be a 2-carbon chain, so we've broken down from our 3-carbon pyruvate. And what happens, the big idea, this is all I want you guys to get, so you don't have to write this whole thing down, is notice that as we move along, we continue to break down that chain. And every time we break off a carbon, we release CO2, because there's that carbon that's being released from here. Also notice that when we break off a carbon, we've broken a bond, so we've got a little bit of extra energy. So what are we going to do? We're going to store that energy in either a high energy hydrogen bond, like we did right here. So I'm following around this circle. I get citric acid to isocitric acid. I see my CO2 being released, and then I see my NAD plus that's willing to take on a hydrogen, and it's going to pick up a hydrogen that's going to carry to the electron transport chain. If we keep going around, we notice that in addition to storing hydrogens, high energy bonds, we're also going to make some ATP. And so this cycle will help us to store energy, and hydrogen bonds also produce ATP. You are not responsible for memorizing the steps in this, just the big idea. I want you to understand what's going on. The last thing we have that happens in cellular respiration is called the electron transport chain, which is often shortened just to ETC. So at this point, what's happened is we've stored a lot of energy in these high energy bonds, these hydrogens that we've stuck in on the end of NAD and FAD. What's going to happen is these hydrogens are going to build up across the membrane of the cristae. So we're going to put a lot of positive hydrogen atoms on one side of the membrane. And I'll show you guys what this looks like in a second. Now, remember from chemistry, when you get a lot of molecules that have the same charge on one side, they don't like being near each other. Opposite charges attract. So what's going to happen is these hydrogen ions, the more you pack them across the membrane together, the unhappier they're going to get being near each other. What they have are these channels, so these proteins that are stuck in the membrane. What these channels are is they're a way for the hydrogen ions to get away from each other. So as the hydrogen ions rush through these channels, they're rushing through with such force that their movement through the channel is actually going to make ATP. And this movement across, for every 1 molecule of glucose that we take in, we can actually make enough hydrogen ions through NAD and FAD that we can make 32 ATP in ideal conditions. So here's what that looks like. So your electrons, so FADH and NADH, they're going to drop off their hydrogen atoms, and they're going to get pushed across the membrane. So they're going to use the movement of electrons is going to act as a pump to move these hydrogen ions across. Then we build up a huge quantity of hydrogen ions on this side, and they are all similarly charged. They're all crowded in there, so they are very unhappy being over here. They don't want to be stuck on this side of the membrane together. They want to get on the other side. But there's only one way to get across, and that's this bright pink protein right here. So as they rush through right there, they do it with so much force that that force actually turns ADP, so adenosine diphosphate with 2 phosphates on it, into ATP. So it stores energy from that movement across the membrane in that bond right there that can then be used by other parts of the cell as an energy currency. All right, so quick review. You should have your notes filled out. Make sure you go back and you diagram the parts. So diagram your parts of cellular respiration where they occur. Also make sure that you have your parts of the mitochondria. Do a little bit of review, and if you have any questions, please feel free to ask.